So the Ames test is this really cool way of deciding if a chemical that you have is a mutagen. And what we do with your suspected mutagen is we expose a single-celled prokaryote to the mutagen, and we see if the mutation rate changes. And the reason we can do this, right, is because our single-celled organism has a really short generation time, and they're going to divide and have a ton of replication events over a short amount of time. And every time there's a replication event, there's the chance that a mutation will occur by chance. So there's already a background level of mutation existing in your population. If you expose that same population to a mutagen, what you would expect, your hypothesis is that the mutation rate would increase. You would see more mutants in that population with the mutagen than you would see in the population without the mutagen. You're still going to see mutants in both populations. And so that's an important idea to keep in mind when you think about the Ames test. And so what's being shown here in the image is, your two petri dishes that have your salmonella, in this case, typhimurium is our study species. And this particular strain of salmonella cannot synthesize histidine. So, the majority of the bacteria that are put onto the plate will not be able to survive because they're not able to synthesize histidine and they're not able to grow on the medium. You expose that population to rat liver extract, which has histidine in it, and some of those mutants will take up the ability to make histidine from the rat liver extract, right? So some colonies will grow on your petri dish, and those are your mutant colonies. Those are the ones that, because of their exposure to the rat liver extract, they were able to, by random chance, have a mutation that landed in the location of the histine controlling gene, and that caused a point mutation that led to them being able to produce histidine. Okay. Then you do the same thing, but you expose them to your suspected mutagens. So you could do it with cigarette smoke. You could do it with a particular solution that you are worried about. And you see how many colonies grow in that petri dish. More colonies grow in the suspected mutagen experiment, then you know that additional set of mutants was due to an increased mutation rate in that new population. Which you can attribute to your mutagen. So I think Ames tests are pretty cool. Because we have them, we have been able to actually run Ames tests with cigarette smoke. So we're able to actually expose, microbes to cigarette smoke, and we're able to measure if their mutation rate increases or decreases over time as a result of the exposure. And every single time, right, mutation rate increases. So we are confident that cigarette smoke is a mutagen that has a lot of effects in the cell. This slide is just illustrating some of the warning labels that we now put on cigarette packages as a result of understanding of how mutagens kind of work. And so if you think back to the first week of class, first lecture, biology affects our society. This is an example of one of the positive effects of biology on society. These are cigarette labels, from 3 different countries. And there's been a consensus. We now have a consensus worldwide that smoking increases your exposure to a harmful mutagen.